So welcome to our very first virtual PLEON workshop. My name is Beth Norman. For those of you who don't know me, I'm the Director of Science and Research at Lacawac Sanctuary. I'm also the Director of the PLEON program. So I wanted to go quickly over some Zoom technical information. We're all probably getting very familiar with this particular platform.
Yeah, welcome everybody to the, as she said, the first virtual workshop that we've had with this harmful algal blooms in the Poconos. This is part of a series of workshops in the summer that are provided for free by PLEON, which is the Pocono Lake Ecological Observatory Network. And I'll run through some of the goals of PLEON. It is based out of Lacawac Sanctuary and Biological Field Station in Lake Ariel, Northeast Pennsylvania. And the goals of PLEON are to empower the public. So we're doing a lot of workshops, helping to inform, as well as meet the wonderful individuals that are involved in the lake associations and the lakes themselves to kind of help better understand and manage the freshwaters that they live by and love. It also helps create a community of scientists, students, environmental educators, and landowners. So we're just trying to bring everybody together so that we can talk and converse about, the issues or challenges, as well as the benefits and exciting news related to lakes. Myself, I am one of the scientists, students from Miami University, and we have a fellow the PLEON intern, Daniel Kaufman, who's from Gannon University. And then another part is providing and communicating the data. So PLEON goes out and samples lakes, and the whole goal is to get a better idea of what's happening, not only in the short term, but long term of the lakes in our community, and to ensure that landowners fully understand some of the advantages and limitations for different lake management approaches.
Hi, Lauren. Sorry, I got out there.
Okay, there's
you did a great job. Good for starting.
Okay. I will give it back to Beth. I ran through just these 3 points. So
perfect. Thank you so much. And I heard you introduce Daniel. So if you have any questions as the workshop moves forward, feel free to use your chat window, and Daniel will be monitoring that chat and make sure that the questions get to Lauren. You can also wave your hand, and Daniel will make sure that you're called on and that Lauren is able to answer your questions. Okay, so Lauren did a great job introducing PLEON, so now hopefully I will be able to introduce her. As she said, Lauren is a graduate student, a PhD student at Miami University of Ohio. She has a master's in public health from University of Tennessee. And she has been studying lake browning in the Poconos, as well as harmful algae and what might cause harmful algae, and the different types of ways we detect harmful algae, specifically in Pocono lakes. She's been working in the Poconos since 2016, and she's been a really valuable part of the PLEON Science Advisory Committee. So with that, will turn it over to Lauren and hopefully be able to stay with you the rest of the time here.
Great, thank you, Beth. So you probably may have seen or heard about harmful algal blooms, maybe from a friend or in the news. Here where I'm based in Ohio, we've dealt with harmful algal blooms for a long time, especially in Lake Erie. You may have heard about in 2014 when Toledo had to shut down its water source for over 500,000 residents due to a very toxic algal bloom that occurred. And Daniel lived with that while that was going on. And when I started this workshop in 2018, it was happening at the same time that there was a big mass toxic algal bloom occurring in the Gulf of Mexico called the red tide, in addition to a blue-green algae occurring in Lake Okeechobee that was draining towards the Gulf of Mexico, and it was causing a lot of en masse die-off, fish kills, and some health effects from that. Last year, 2019, had a very good year for toxic algal blooms in the northeast region. And so we just wanted to put together this workshop to kind of help explain what harmful algae are and what you need to know related to your lakes. There is some question as to whether harmful algal blooms have been increasing more recently, or if we're just noticing them more. So this is a map of all the confirmed harmful algal blooms across the continental US, and they have been found in every single state, but we're focusing mostly on Northeast Pennsylvania and what you may find or experience in your lakes. The first question of the day: what are harmful algal blooms? So I just want to talk a little bit about the different definitions here. So algae are non-vascular plant-like organisms that live, they can live in terrestrial environments as well as aquatic environments, but they're mostly defined by their ability to photosynthesize or make their own food using light energy. We're focusing today on those that live in aquatic systems, in particular the ones that are free floating, so they're not attached to anything. And their more scientific name is called phytoplankton. And I just want to point out that they're a very diverse group. We're talking about not one single organism, but many organisms that live in this community. They can vary in size and shapes, cell shapes, as well as they can just be free floating as single individual cells, or aggregate as colonies, or develop as filaments, as you see in some of the images here down on the right. They can come in all colors as well, and their taxonomic groups are named primarily based off of the colors that they produce. For example, the green algae or the chlorophyta have the pigment chlorophyll, which produces that green color that you see. There are cyanobacteria, or the blue-green algae, which you'll hear more about in this talk, which have the pigment phycocyanin, and so on and so forth. One I wanted to point out real quick is the image here of the little dinoflagellate. This is Carina brevis, which is a marine algae, or alga, I should say, but this is the one that's responsible for the red tide. But the rest that we're going to focus on today are freshwater phytoplankton. I want to point out that phytoplankton are living in the aquatic environments all the time. They're there. And it's very rare that you see a bloom, as well as even more rare that they become toxic. So we're going to talk about a little bit on the fact that they're very dynamic. The population of these organisms is very dynamic. When we talk about why they grow or developed, you can approach it in two ways. One is this bottom-up concept, in which the populations are limited by their resources, so light and nutrients they need to survive. You can also approach it a different way related to what is driving the populations down, which is more of a top-down effect. So the phytoplankton themselves are the very base foundation of the food web in aquatic environments such as the lakes that you live near. And a lot of times their populations are dependent on the edibility of the phytoplankton themselves, as well as consumer preference, who likes to eat them. And this wouldn't be a Williamson lab presentation without an image of the cute little Daphnia zooplankton that feeds on algae. Their common name is the water flea, but I like to call them the water cows because they graze all day long. Another point I wanted to mention is that these populations are very dynamic in that they shift in total population, as well as who's dominant at the time over both space and time. For example, in this image down below is from the phytoplankton ecology group that developed a model that kind of generalizes the typical pattern you'll see in a temperate region. So you'll see a high increase in the algae in the springtime. You may notice a bloom just simply from all that spring showers bringing in nutrients. The lakes are starting to warm up, and so they become very productive. Then it's followed by a graze down or bottom down top-down effect from the zooplankton that are eating on the yummy algae, causing a clear water phase, so the lakes tend to clear up. And then all that's left for the summer is typically cyanobacteria that will then bloom in midsummer that you'll see and we'll talk more about later. I also want to point out that although most of the time algae are very beneficial and they're the base of the food chain in lakes, but they can also be detrimental at high densities, so very dense populations. There's two different ways you can describe a bloom. One in particular, which we'll focus on, are toxic blooms. These are the phytoplankton populations that are releasing toxins when the cell breaks open. And these toxins can either kill or injure aquatic and terrestrial organisms that come into close contact with the water, swallow it, or even inhale it in a wave spray. But most of the time, what you'll see are non-toxic blooms. These are more of a nuisance in that they can be aesthetically unpleasant. They can lead to odor and taste unpleasantness of the water, as well as boating or hindrance. If you've been out during a bloom, you may notice it's harder for your motor to run because the algae can clog, filters. They can clog intakes for water treatment plants, as well as some fish gills, and can cause some damage that way. But mostly when you see a fish kill or en masse die off of fish in the lake is typically because the bloom has already died and settled to the bottom and it's being decomposed. And all that decomposers are sucking out the oxygen, which is none left for the fish to survive.
Lauren?
Yes.
So aside from the fish kills, when the bloom is decomposing, does the algae help support fish in the lake in any other way? Or is it totally detrimental?
Yes, it's when they're not very dense. So when they're at normal populations, they are, the zooplankton that feed off of the fish are the primary source for juvenile fish. So they are absolutely necessary to have fish in your lakes. There are also some fish, such as shad, that feed directly on phytoplankton. So they're most of the time, very beneficial for the lakes. It's only on these rare occurrences that you get such dense populations that they can become detrimental.
Thank you.
Yep, good question. So this comes down to, what are the causes of these harmful algal blooms in freshwaters? And in freshwater, we're particularly talking about cyanobacteria. So these are the blue-green algae that you may have heard about. They're very ancient. They arrived in the fossil record we've seen dating back all the way to approximately 3 billion years ago. They evolved under very extreme conditions. They are considered the original origin of modern plants, as well as oxygen in the atmosphere. So another benefit of algae is you can thank them for the ability of us to breathe on Earth. They are, on the flip side of that, they are great competitors because they evolved in such extreme conditions. They can rapidly multiply. The record was 2.5 hours to double their population. They have a higher efficiency for nutrient uptake, so when nutrients become very limited, they're the first ones to get the nutrients. And they grow better at higher temperatures. So when the lakes, later in the summer, when it's very warm, above 25 degrees Celsius, you'll start to see them really enjoying themselves and overrunning the other algae. There are some that can control their buoyancy. They have adapted gas specules that help them to control where they are in the water column. So that can help them optimize light capture and nutrient capture. And on another side of it, there's some pesky ones that can switch to consuming organic matter when they can't get nutrients the normal way. So in the image to the right is just showing the different types of algae or phytoplankton. And most of the benign chlorophytes, cyanoflagellates, diatoms typically have a lower peak optimal growth temperature, and cyanobacteria, they just love the warmer temperatures. So that brings us to the toxins that cyanobacteria can produce. These are secondary metabolites, so they don't need them to survive. The million-dollar question is determining why they or how they use these secondary metabolites. There's some theories as to whether they use it for protection against UV damage or to just simply store excess nutrients. But the billion-dollar question is what causes the cyanobacteria to switch to toxin production? And these two causes are still yet to be determined. I want to point out that some species can produce more than one toxin, and there are some toxins that are produced by many species. So again, they're very dynamic. When we talk about the toxins, there are generally three types. One is a dermatoxin, which is where you can get inflammation of the skin, eye, just from being in direct contact. There's hepatotoxins or liver toxins.
Regular.
These are the microcystin, nodularin, selenospiropsin. They can cause respiratory distress, gastrointestinal distress, headaches, fever. And then the more severe toxins, the neurotoxins, can even lead to neurological damage. And I want to point out, you may have, the picture to the right is of the three dogs, Abby, Izzy, and Harpo, who perished last year from just a couple of hours exposure to cyanotoxic bloom in a pond in North Carolina that made massive news on social media.
Lauren?
Yes.
This kind of also goes hand in hand with the question that we have about the safety of swimming in water with algal blooms.
Yes, so the standard recommendation is if there's a bloom, we don't know whether toxins are present, so it is recommended to stay out. There are, and I can get to the, I will be touching on this a little bit later in the presentation, but as I mentioned earlier, most of the time, toxins are very rare to occur. But the standard recommendation is if you do see a bloom, it's recommended to stay out of the water and keep your pets away. I have a little asterisk by saxitoxin because that is the only toxin that's federally regulated because it was developed as a biological warfare agent by the Department of Defense. So you may be wondering, you know, when do toxic blooms typically happen? Again, they're very rare, but they typically occur when there's high nutrients available. There's some, you know, argument as to which one's more important, nitrogen or phosphorus, but we've really found through the literature that it's, you know, both are very important, as well as what forms they come in. Warmer waters, again, because cyanobacteria grow so well in warm water. More of these stagnant or slow flowing waters, simply because they can control their position in the lake to get the nutrients they need or the light availability. That typically happens a post-rain event. And relatively shallow near-shore waters. You'll typically see cyanobacteria blooms will appear as pea soup or floating dots or clumps, or it may look like someone spilt a paint of, you know, green paint can in the lake. Another one is if they look like green streaks, like somebody threw their grass clippings under the water. And this image here of the right is showing that of a typical cyanobacteria bloom we've seen in the Pocono lakes, where it looks like grass clippings. So I'll pause for just a second if there's any questions particular to the previous slides.
Lauren, I have a quick question. When I go down to the lake right now, the temperature is like 72 degrees, and there's these very small molecules, I don't know how to describe them, green, that are floating all in the top 1 foot of the water. That doesn't mean anything, so can you explain what those might be?
So it's hard for me to determine for sure what they are. But typically if it's, I would say if it's green clusters floating in the water, it sounds like algae, but we don't know what type of algae it could be without looking at it under the microscope. So seeing it could be a very, you know, benign green algae doing their thing, or cyanobacteria, and there's many species that can either produce toxins or not. So it's really unsure without looking what it could possibly be.
You can also use the chat then.
But an image here, I'll say, is a classic green algae bloom that we've seen in the Poconos.
Lauren, it's Pat Stevens. I just have a quick question. Does the Lacawac Sanctuary, if we bring a sample of the algae to you, could you determine what it is?
Yeah, so we're gonna get in, that's a very good question. We're gonna get into that in a little bit, but because PLEON has a monitoring plan for Pocono lakes, which I'll leave Beth to answer. So very good question, but I'll leave Beth to answer that here in just a little bit. But at Lacawac Sanctuary, they do have the equipment to look at the algae, but again, I'll leave that to Beth here in a little bit because she'll go into all of that.
Lauren, I have a question.
Yes.
This photo is,
Well, you type it in.
it looks like the blue-green algae that we get on our lake later in the year, and like later in August. Is that blue-green algae?
Yes, this was a confirmed blue-green algae bloom. Yes.
Okay. So, but
again, it's hard to tell. And usually when we have this workshop in person, I show a lot of images just that look relatively similar to see if you can determine. And the answer is you can't determine what it is just by looking at it.
Okay.
I can't confirm through this image that it was a cyanobacterial bloom.
Okay. I will.
The next part of the slides are going into some common challenges and best practice with monitoring, detecting harmful algae. So I can move on to that if a lot of the questions are related.
Lauren, we do have a couple other questions here. I believe Pat was waiting to ask a question. And also, you know, there was a question of is there a way to visibly tell whether an algal bloom is toxic?
To physically, without running a toxin test, no.
Okay. And then Pat did ask his question of how much of the scattered paint algae is needed to keep people from swimming. So is there like a threshold limit, a visible threshold limit?
So that is a very good question, and one that I was going to bring up in a little bit. It's kind of the wild west right now with thresholds, in that there's no federal standard or threshold. There are recommended thresholds that EPA has developed based off of information from the World Health Organization, as well as other agencies. And I'll get into that in just a little bit, but it varies depending on what method you use. For example, a visual method, if you're looking at whether a bloom is present or not, there are visual scores you can give. If you're sampling for how much total algae are in there, it's typically, if there's 10 microgram per liter of phytoplankton, it's considered a bloom. And there's also a way to detect them via visual fluorescence signatures, and that I'll get into in just a little bit. But yeah, very good questions. Again, going into challenges with HAB monitoring. Blooms can be very dynamic, as I mentioned before. The algal population.
She's got to get back to it.
Lauren, can I just make a statement? Could we please ensure that everyone is muted if they're not asking a question, because we're getting a little bit of feedback.
Okay.
Thank you.
So blooms are dynamic. They can develop very quickly within hours, and some appear out of nowhere. They're not appearing out of nowhere. They do double their population relatively quickly, but most of the time they're hiding deeper in the water column and they float to the surface, which makes them appear so quickly. So we consider them hiding more in the deep waters. And they can change their location via the wind. Once they reach the surface, the wind can blow them and concentrate them in a particular localized area, or they can be very general and cover the entire lake. One of the challenges is the visual confirmation of bloom does not always indicate toxins are present or at harmful levels. And as I mentioned before, through very widespread sampling, a toxin event is very, very rare. Toxin producing genes may be present or absent even within the same species, and a single toxin producing algae can split between toxin-producing and non-producing stages. Again, going back to that billion-dollar question. I also want to point out is that the lack of a bloom does not always indicate toxins are absent. For example, toxins are retained within the cell, so only when the cell is broken, either from being eaten or lysed after the cell dies, that the toxins are released. And they're normally biodegradable, and they will degrade naturally in the water. So typically the standard is to wait until the toxins decrease. But some of them can be a little bit more resistant to degradation, so they can persist in the water up to several weeks. And the other key challenge is, at this point in time, toxins analysis is very expensive. And most of the toxin tests only test for one, either microcystin or cylindrospermopsin or anatoxin. And they're run about several $100s per sample to test. And as I mentioned before, you may know that certain taxa produce more than one toxin, and there are different, up to, microcystin has over 80 variants of its toxin, and the test only sample for a handful of them.
So Lauren, does this mean that there are there any like small test kits that citizen scientists can use, or does this all have to go to the lab?
That's a good question, leading into my next slide. So in general, there are two types of toxin tests. One that's recently been developed, which is the Abraxas quick dip test strips that we've used in the past. And I want to point out that they're qualitative in that they'll tell you presence, absence, and a general range, but they're not the end-all, be-all, and they only test for one toxin at a time. So you can run the samples. A pack of 20 of them costs about $500. So you can run 20 samples, but they only test for one toxin. The other option is to take a water sample in the lab and run a similar test, which is just as expensive, as well as if you perform liquid chromatography, mass spectrometry to look at what all the molecules are present in the water. That is also very expensive and time consuming. So if you're looking for a more rapid, quick test, the quick dip test strips, you get your answer within, an hour, 30 to 40 minutes, whereas the in-lab procedures take at least 7 to 8 hours. There are other ways that, because the toxin analysis are very expensive, and you talked about different threshold ways in which we can detect a bloom. One of the methods, the most commonly used method, is just visually. You can take a sample, put it in a bottle of water, and if you see them floating, all the algae floating to the surface, that means that they have gas vacuoles, so they're more likely cyanobacteria. Other types of algae will just simply settle down to the bottom. We can use sensors that measure their pigment fluorescence, so the color of them, to determine what type of bacteria they are, as well as get an idea for how much phytoplankton there are. And this method has been used all the way from a portable sensor, just like the C6 in the middle image, as well as those that are used on the satellites orbiting the Earth. But those typically can only measure in the larger lakes, such as the Great Lakes. The third way is to grab a sample and identify what's there. You can look at the taxa via just the microscope, or send it off for genetic testing to see if they have the toxin-producing genes. I do want to touch base a little bit on what are the most successful strategies for detecting and responding to harmful algae for those who may be part of lake association management. The most successful strategies all have these 4 points part of their plan. One is educational outreach, such as what we're doing today, and helping to train citizen science to do those more in-field testing that can be done relatively easily and with little training. Including a routine monitoring plan with the lake. As I mentioned, populations are very dynamic. They can come and go relatively quickly. And then having an incident checklist, so if you were to detect a bloom, what is the next steps? For example, if you have a visual detection, do you go to fluorescence or do you send a sample straight to the lab? These are all, again, decisions that are not regulated or required. There are procedures or protocols that have been put together for associations to use or to adopt or to, so again, this is all on, in Pennsylvania specifically, they don't have required or routine monitoring for harmful algal blooms. So that falls on the shoulders of the individual lake associations. So it's important to have a HAB management team, a response team, or even a point of contact in case a bloom is detected. And one key point that we practice as well is this cost share program and or state federal partnerships, because the toxin testing or sending samples to lab can get very expensive, especially if you're doing on a routine basis. So helping to share that burden is been very successful. This image down to the right, I just want to point out, is not Pennsylvania, it is Kansas, but they incorporate routine visual detections for harmful algal blooms in their public waters as part of their everyday traffic notifications. So, and their HAB strategy is posted on their state website. So what are some things that you can do to help manage harmful algal blooms or help control or reduce their occurrence? Some successful strategies have included putting in wetland buffers around the lake to help control the nutrients coming in, reducing your fertilizer applications on the lake properties so that you're not adding more nutrients and feeding those cyanobacteria in the summertime, upgrading or improving septic systems because they can, septic discharge can be high in phosphorus, which is the key limiting factor, one of the key limiting factors for cyanobacteria, as well as controlling any livestock or waterfowl that may be close to the lakes and contributing to the nutrients. There are some industrial chemicals or bacteria, enzymes, products that you can put in the water. But again, you have to be very careful with that, in that you don't want to kill the good algae, because then all that's left is those potentially harmful ones. Aerating of the deep waters has been used successfully to help reduce that regeneration of phosphorus from the deep sediments that cyanobacteria go down and scavenge for. And then some help with barley straw for erosion control and stabilizing banks has been used. And then I've provided some local resources, and I do want to point out there's no statewide program right now for harmful algal blooms. It's all falling on the shoulders of the lake associations, but there are resources available, and the state is in the process of developing a program or a task force to handle harmful algae in Pennsylvania. And here are some key people to contact: Brian Swistock, who's from Pennsylvania State Extension Office, and Peter Wolfhouse, who handles the community side of harmful algal response. Beth Norman from PLEON, and you can report blooms directly to the Pennsylvania Department of Environmental Protection. And with that, I will hand it over to Beth.
Lauren,
First I have questions, yeah.
Yeah, we have a few questions. If the individual that has the iPhone would like to take this moment to ask a question.
You may be muted.
Yeah. And Pat also has a question here as well.
So the question I have is, you mentioned something about greenscaping. Can you go into more detail about what that means?
So greenscaping is an idea that comes more from helping to reduce impervious surfaces in urban areas, but it's also been found to help with reducing nutrient loading to lakes, because if you're reducing the amount of impervious surfaces by adding more, rain gardens or less concrete, surrounding in the lake watershed, then that's less runoff, which during a rain event, and the runoff is what's bringing a lot of nutrients to the lakes themselves. And, you know, you can do some greenscaping or, you know, rain gardens or, as I mentioned, some barley straw or controlling erosions around the lake itself to help capture some of those nutrients before they go into the lake.
Okay, let's see if we can get the individual from iPhone back on here.
This is Chaz Ritson from Birchwood Lakes. Can you hear me?
Yes.
Yes.
I wanted to ask one question, but I wanted to make a comment about the earlier question about seeing something on Lake Wallenpaupack. This time of year, it's very typical to see pollen on the water, and it's easy to confuse the issues between pollen and algae. I just wanted to mention that because at all of our lakes this time of year, when you see your cars turning green, that pollen would also be in the water. Okay.
Absolutely. Yep.
I just wanted to ask a question. I know you went over this, but it sounds as if there is no new developments on rapid testing other than what was discussed last year at this time.
Correct. We have the quick dip test strips, the classic methods that we've described before. There haven't been any, there are some that have been developed or in development but haven't been pushed or accepted as, what do I want to say, dependable measures for toxin analysis yet.
Okay, thank you very much.
Lauren, we also have several other questions here. We'll start with this one. Someone is asking about testing on Lake Wallenpaupack. Are they testing for toxins? Are they concerned about the increase in algae? And does holding the water at a higher level contribute or not contribute to the increase in algae?
So I would have to say, related to that, I myself have not tested with Lake Wallenpaupack. PLEON is not monitoring Lake Wallenpaupack except for individuals who have wanted PLEON to come and sample off of their dock. But PLEON is not involved in the routine monitoring at Lake Wallenpaupack. That is performed by the Lake Wallenpaupack Management District. And to my knowledge, they do not do routine toxin analysis. But I'll have to refer you to the Lake Wallenpaupack Management District point of contact for what they include as far as and what they have tested in Lake Wallenpaupack itself.
And Lauren, what nutrients or minerals should be monitored in the lakes to determine if there's a potential problem with algae?
So related to the nutrients, the two key factors are nitrogen and phosphorus. So there is, as I mentioned before, there's some discrepancy as to which one's more important. The answer is they're both important. And you really have to do sampling within your lake because lakes, even in the same neighborhood, could have completely different watersheds and nutrient limitations. So it's important to look at which nutrient is more abundant, as well as what type of algae are proliferating in that lake to determine the answer to that question. I do want to point out with cyanobacteria, and I'll go back a little bit here, there are some cyanobacteria that have specialized cells that allow them to fix their own nitrogen from the atmosphere. So controlling nitrogen may not help with those populations. The more limiting nutrient for them is controlling phosphorus.
And how important is it to control grass clippings or leaves from being discarded into the lake?
How typical or how important it is?
It's how important is the question. Is it to control grass clippings or leaves from being discarded in the lake?
So the grass clippings, I would say, is more related to them being a source for the heterotrophs or the bacteria that decompose organic matter in the lake. And so those are what's going to be decomposing the grass and releasing the nutrients that could be gained from the grass clippings. More importantly, is the controlling of the spraying of fertilizers to keep the grasses green. After a rain event, if you spray before a rain event, then those nutrients that weren't used are going straight into the lake and can feed the phytoplankton. So it's not really the grass clippings themselves, rather the nutrients that may be sprayed on them or that can be generated from when they're decomposed at the bottom of the lake.
We also have a question from Paula and Mike Vorak. If you would like to state your question now, please.
You may be on mute.
No, can you hear me? I unmuted.
Yep. Yep.
Okay. I think my question was just partially answered, but if originally was, if there was a toxic bloom on Lake Wallenpaupack, you know, who would determine that, and then how would that information be communicated?
So again, I would have to defer to Lake Wallenpaupack Management District and their progress on a HAB management strategy. As I mentioned, there is no state regulations for monitoring or reporting toxic blooms at this point in time. I do know that they are working on that and developing a task force to put together some requirements or recommendations. But right now it all falls on the individuals or the lake associations that are in charge of monitoring the lake themselves. So, yep, it's kind of the wild, wild west right now related to response strategies. And, you know, I don't want to speak for Beth and for PLEON, but when PLEON monitors, those results from that are solely the property of who we collected the data from off their private dock or that lake association.
Lauren, is there a website where that updates the water quality on lakes all across the country that people can go to and see what's going on and make maybe some comparisons?
Yeah, so there's a couple of resources there that have been developed for that in particular. One that the EPA, Hillary Swook, who is at EPA, he and his team developed it's a mobile app for Android phones. I believe you can use it on iPhones now, but it was originally developed for Android phones. It's an app called Bloom Watch, where you can go and look at you know, if there's an algal bloom right now happening in your pond or your lake. And it also, you can upload data, you know, if you take a secchi depth, how clear the water is, you know, the temperature of the lake, that can be added on there and can be viewed by the public. There is a water quality watch website that you can go to through the US EPA. That is citizen science, and trying to think of any others. Those are the two main ones that I can think of. Beth, do you?
No, the two main ones, and I would emphasize that they're not comprehensive. So those are the blooms that citizen science report. You know, just regular people report. It doesn't mean, if you don't find your lake on that list, that doesn't mean you don't have a bloom. It just means nobody has put it into the system. So there, especially in Pennsylvania, specifically, there's no monitoring statewide. So it's kind of just people reporting what they see. It's not any kind of state-led initiative at this point.
Lauren, what are the threshold levels of phosphorus and nitrogen? What would you say are the levels of concern when they reach a level of concern?
So again, that's more related to a lake, to the specific lake. So some lakes that are naturally more green, have more nutrients, well nourished, they naturally have more phytoplankton in general. And so they can have much higher total nitrogen, total phosphorus concentrations versus the more clear lakes, blue lakes that typically have less nitrogen and phosphorus during normal times. A very small change can cause a great response in that lake versus a small change in a greener lake. So that speaks to the importance of monitoring for nutrients and getting a baseline for what's natural, what's normal, I shouldn't say natural, what's normal in your lake. And that way you can determine whether, you know, at what point do the total nutrients will lead to a response. But again, there's not one single number that I could speak to because lakes are so diverse.
And could you reiterate a little bit on how the barley straw is used and how effective it may be for small lakes?
So the barley straw has been used typically for erosion control, but has been shown to help absorb some of the nitrogen and phosphorus in runoff from precipitation. I cannot speak to it specifically since I have not used it, but Peter Wolfhouse is the point of contact for that, I would say, who has used it and has monitored its effectiveness.
This is a very good question, especially coming from someone here. Living in Erie, we have geese everywhere, and we have a question of how much of a factor does goose or geese fecal matter play in feeding algal blooms?
Oh yes, this is a kind of a fun question in that this does depend on the size and depth of your lake. But in more shallow, smaller lakes and ponds, the geese can have a significant impact. And there's some great photos, just Googling it, before and after geese have passed through during their normal trips, you know, north and south. But in larger lakes or deeper lakes, again, this points to you have to look at how much nutrients are in your lake on a normal period, and whether it's normally low nourished or poorly nourished lake, then certainly a big gaggle of geese coming in and providing that yummy nitrogen can make a big difference in those types of lakes. But typically those greener lakes, not as much of an issue.
Unless anyone has any questions, we may be good on moving forward.
Okay, so I think that's my cue. So I wanted to talk a little bit about PLEON, specifically what we can offer or how we can help you think about monitoring HABs and taking the information that Lauren provided you today and doing something on your lake. So I'm going to talk about 3 things that PLEON can help you with, or services that we have. First of all, we do have a formal HABs monitoring protocol. So this is typically, PLEON will work with lake associations to develop a sampling regimen. And this sampling regimen can include a lot of things. We measure lots of different water quality variables, including nitrogen and phosphorus. So some of the trends that Lauren was talking about that might be important for you to know on your lake, you know, how much nitrogen do you have? How much phosphorus do you have? Is that changing over time? PLEON monitoring can help you answer that question. We create basically a record of your lake and how it might be changing over time. Specifically for HABs, we do a 2-step screen or a 2-step HABs monitoring process. The first thing is called a PTOX screen, or potentially toxigenic cyanobacteria screen. So if Lake Association wanted this service, what we would do is we come out and we take 2 water samples from your lake, 2 different locations. Usually I do like a center lake and then one closer to your docks. And we take those samples and we send them to a lab that we work with in Florida called Greenwater Laboratories. And Greenwater Laboratories is a specialized lab that has expertise in algae identification. So they take that water sample, they look at the algae in it using a microscope, a specialized microscope, and they identify what species are present. And specifically, they're looking to see if the taxa that we know are capable of producing toxins are present. Okay, so the key with this is that there's a very quick turnaround time for this service. So Usually you get your results back within 48 hours of me shipping the sample to Greenwater Labs. So the reason we do this initial screen is that if they do not find the type of algae that we know can produce toxins, if they are not in your water, then this is a good news, right? If they are present in that sample, depending upon their abundance, Greenwater Labs may recommend testing for the actual toxins that Lauren was talking about. And they can narrow down that really wide list of over 40 different toxins to a narrower subset based on the taxa that they found in that sample, because we know which algae are capable of producing which toxins. So that's the second step, is the quantification of the actual toxins, where the lab will do a laboratory analysis to see what the concentration of those specific toxins are in your water. Again, this has a really quick turnaround time. So what you want is to know if the toxins if you have toxins in your lake, you know, during the summer. You want to know when you're going in the water. You don't want to know months down the road. You want to know right away whether your water is safe for you to go into or not. So that's the point of this formal HABs monitoring. PLEON reports those results directly to the Lake Association that we have agreements with. We do not make them public, and we are not implementing a response plan. PLEON does not shut down your lake. PLEON does not tell you to shut down your lake. PLEON does not tell you you should not go in the water. PLEON simply says, these are the results, and we can help you understand those results, but it is up to the Lake Association to make a determination as to what to do or what action to take or not to take based on those results. The cost for our PLEON service, that formal HABs monitoring, is about $400. And if you're interested in that, just contact me and we can work together to design a sampling regimen for HABs or for any of the other water quality measurements that PLEON does. Okay, Lauren, if you want to go to the next slide. The other way that PLEON can help you is with our citizen-led monitoring. So we have a citizen science program that is designed for individuals. So our formal HABs monitoring is usually between PLEON and a lake association. Our citizen science program, we gear towards individual lake enthusiasts or lake owners. So what we have is a lakeside monitoring protocol that it is designed, again, for the individual to do. This protocol includes a bunch of different water quality measurements that are very simple to do, but very, very powerful, give you good information. One of those variables is a visual assessment of the algae in your lake. And I'm showing you a picture there of what that protocol looks like for algae. So what we help you do as a citizen scientist is to evaluate visually the algae that's around your dock or your sampling location. And we do that periodically through the summer. That's the way the program is set up. So what you're starting to do is develop a record of what your algae looks like over time in your dock or your shoreline. This is a visual assessment. It does not include identification of the algae or of the toxins. Okay, this is just a way for you to get an idea of what areas might be problematic. Okay, we're not telling you that there's toxins. We're not telling you that there's not toxins. We're not even telling you what kind of algae it is. We can only visually assess what it looks like. This works best when you have lots of people working on the same lake. So depending upon the size of your lake, if you get 4 or 5 or 6 of your neighbors all around your shoreline to do this all together, you can really start developing a nice picture of what your lake is doing over time. The cost for this, we ask a $50 donation for the kit. That basically just covers some of the sample analysis that happens with some of the other water quality parameters. You can look at our website for training, our training schedules. You do take about an hour training. You get your kit, we go through all of that. But if you're interested in that, let me know or look at our website for the upcoming training dates. Okay, Lauren, one more. The other thing that PLEON is working on now, so we are working with the Department of Environmental Safety or Environmental Protection in Pennsylvania. So the PADEP does not have a formal monitoring protocol or system, but they are starting to do these surveys to get an idea of harmful algae in Pennsylvania. This is very informal. This is basically one guy at DEP who has some money to do this kind of work. PLEON is working with this contact at DEP in order to provide samples from the Pocono region. So what we do is basically, if you think you have a HAB or if you have an algal bloom that you want to know more about, you can contact me. I'll come out, or PLEON will come out and take a sample, much as we do for our formal monitoring program. I then send those samples to the DEP. And at that point, the DEP they look at the algae using a microscope with the various labs that they work with. They do the P-tox screen, they do the cyanotoxin quantification, they do all of that. The good news is all of that is free for you because this is a DEP program, a survey. They have a pot of money that they use to process these samples. The bad news is that there is an incredibly long turnaround time for these data. I did this last summer and I'm still waiting for data for some of my lakes from DEP. This is not meant to tell you what's going on in your lake. It is meant to tell DEP what's going on in lakes all across Pennsylvania. So this is not a way for you to get timely information about a bloom that might be happening. It is a nice way for you to get some free analysis, but it is not going to tell you, again, in a timely manner if your lake is okay to swim in or not. Again, very long turnaround time, although they are working to shorten that. The analysis also depends on funding. This contact has a pot of money. Last year, he ran out. So I sent him samples that ended up not being analyzed because he just ran out of money. So again, this is not really so much for you as it is to give DEP some background information on HABs in the Poconos. But if you're interested, I can certainly come out and take some samples for you. All you have to do is contact me. I get out there as soon as I can. Right at this point in time, they're still setting up the program for this year, so it may be a few weeks. But as the summer goes on, I'll put that information on our website. The other caveat about this program, these data will be publicly available if somebody requests them from DEP. Because DEP is doing the processing, those samples and those data now belong to them. You do get those data, but if somebody wanted to call DEP and say, hey, you know, have you ever done any HABs testing on Lake Lacawac? And you had that done through this program, that information is freely available. They're not publishing it on a website or anything, but you could potentially get it if you wanted to. So that's the other big caveat with this particular program. I think that was all the slides I have. I see a question about students and staff to help me with all of this. Typically I do. Daniel is actually a PLEON intern. This year, however, my interns are working largely remotely. So this particular year, you will likely see me. If you call for PLEON in any capacity, it will likely be me. Hopefully as the summer goes on, depending upon what the COVID situation ends up being, you may see some PLEON interns in person, but right now, this year, it's just me. So that's who you will likely get. So that's what I have for you for PLEON. If there are any questions about PLEON, otherwise I'll turn it back over to Lauren.
And Lauren, we do have a queue of a few questions. Do you want to field them now?
Yeah, we can. I know we're a little bit behind time, but it seems like we have such, good questions and participation. So if you guys are fine with it, we're getting to the last segment of the presentation. So we can do a couple questions now if you like.
Okay, so one question is it safe to eat fish during an algal bloom?
So again, this goes back to whether there's toxins present or not. If you've tested and toxins are present, you don't want to eat the fish that come from the lake. When in doubt, I wouldn't eat. But again, there's no way of knowing without the tests. So.
Okay, and then we also have one here that is, it's a good question too, that when an algal bloom occurs, does it consume the entire water column or is it only present at certain levels?
Oh, I love this question because this is part of my research. The answer is it varies. So I talked about the dynamics of some of these blooms. Some of the populations can be so dense that they take up the entire water column of the lake. Most of the time they occur just within the first couple of meters. Some of them just the first couple of inches when they develop the scombs and float to the surface. It does also depend on how deep your lake is. So if it's a relatively deep lake, it's less likely they're taking up the whole water column. However, for those more clear lakes, you may have some algae, a dense population of algae that are hanging out in the deeper part of the water column just for protection against not only predation, but also UV damage. So yeah, great question. So usually algae are limited to where the light is available.
Okay, and then one more. This is related to a slide that said that PLEON does not implement strategies of response. But the question is, what are some of the potential responses that lake associations or areas could take in order to reduce the effects or the duration of algal blooms?
Yeah, that's a good question. So as Lauren has said, the state of Pennsylvania does not have a statewide harmful algal bloom response strategy, but there are several that exist through other states, and then also Lake Erie. So the Erie County has worked together with some nonprofits and some other area organizations to create a harmful algal bloom strategy, a response strategy. So when somebody asks me about this, I usually refer them to the Lake Erie response, and you can find that online. You can also find it on our website. There's also the state of Ohio has a really nice response strategy. Lauren can probably answer for other states. They all vary, right? So the thresholds are all different across the strategies. What they do at those thresholds are different across the strategies. It really depends, but there are some guidelines for you. There's some scaffolding that you can use to create your own strategy, at least until Pennsylvania moves forward at the state level. But I'll let Lauren elaborate on that if she wants to.
Yes, I would definitely reiterate what Beth said, that there varies across the states because it's simply the wild, wild west right at this point, because there's no federal or state requirements at this point. Vermont, for example, uses visual detection. If there's a visual detection, they will shut down the lake or that segment for a period of time and start doing testing for toxins. I would say most successful strategies incorporate a tiered approach in that there's first a visual detection. They post signage that says a HAB has been detected, it's recommended to stay out. We don't know, if it's toxic or not. Then they either do pigment fluorescence to see what type of algae it is. For example, if it comes up that there's a lot of phycocyanin, a lot of cyanobacteria, they will move to the second tier. If it's mostly green algae or a non-algal, I should mention as well, then they will take down the sign and say it's relatively safe. So the in-field methods are relatively quick. You can get your answer, but again, it doesn't speak to the toxins, but you can get a good idea as to what's there and whether it's potentially toxic. Laboratory analysis, again, I mentioned there is the time that you have to send the samples to the lab. It takes a solid day to run the samples and then another day or two to send you the results. So, and as Beth mentioned, it's highly dependent on how many people are available to do the testing. And most testing, if you run in the lab, is done in a batch sequence. So they're waiting for other samples to fill spots to run the analysis. So to help with the cost burden. So I'll speak to that. And so again, coming back to the question, tiered response, visual detection, typically going to presence or absence of potentially toxic taxa, and then, or pigment fluorescence, and then going to the laboratory testing. And if you're interested, we can send some examples of these HAB management strategies. I would say the National Water Research Council has put together a survey or a report of how well each state does with their HAB monitoring and response, and the top one has consistently been Kansas for their tiered approach, as well as their general acceptance of informing the community and reporting back the data. So I can send, if you want to share your, send an email to Beth or share your email address, I can send you some of those frameworks for good classic HAB strategies.
Lauren, does algae stick around year after year, or you know, how does winter affect it? How does it affect it seasonally?
So this is a good question, and so our slides will be posted at the end of this workshop, so you can go back and look at how they change over the season. But most often they're hanging around year round. Often in the winter time, they will develop, well, during the summer and the fall, they'll develop these actinates or vegetative cells that they will then drop to the sediments and remain dormant over winter. And then when the spring comes around, it mixes, turnovers the lake warms up, you get more nutrients, they start to grow again, and you have a, you know, all new algae there. But there are some that persist year round under the ice. But for the most part, a lot of the algae is relatively dormant over winter. But as I showed before in the image, you know, you'll get periods throughout the year, starting in the spring, where you'll have a high population, then in a clear water phase because they're just eaten up by the zooplankton, and then another high population when you get those summer rains or an input of nutrients in warm waters for the cyanobacteria.
And can you clarify on something? If you put a water sample in a tube, what exactly does that mean for the algae? Will it float or sink?
So this is a visual kind of detection test, in that if you just grab a sample and put it in a cup, it doesn't have to be a tube or in a cup or a glass where you can look at it. The cyanobacteria, several taxa have these gas vesicles that allow them to float to the surface or float to the top. So if they float to the top, then they're more likely cyanobacteria and potentially toxic species. If they sink to the bottom, then they're the benign, you know, diatoms or dinoflagellates, you know, the less likely to be cyanobacteria. It's not 100% foolproof, but it's a quick and dirty field test you can run.
And how do herbicides play a factor?
So herbicides, I don't focus on this part of the research, but from what I've read in talks that I've learned from other scientists who do, is that herbicides, typically their goal is to kill algae. And a lot of times you'll see a nice clear water phase after the algae have died. But remember that cyanobacteria are very competitive, and they're great competitors. And so oftentimes you'll see after the herbicide has worked that the only thing left is cyanobacteria. And you could get worse cyanobacterial blooms after an herbicide treatment because of that. There's simple ecology.
All right, thank you.
Sorry, a longer question, but we have a lot of lily pads on the lake, and I was wondering, does it put the lake more at risk to to let them expand naturally and grow? I mean, they're a beautiful part of our lake, don't get me wrong, but many residents tend to put mats on the lake floor so they do not come up as much, etc. So I just wanted, are we more at risk by having lily pads in the lake?
I didn't hear that last part. Are we more at risk for having lily pads?
Yes.
Oh, lily pads, yes. So lily pads, in that they're petals and flowers, and most of their biomass, it floats at the surface, right? And then they have their stalks that reach the bottom. with lily pads, they're shading that top layer. So they're in a sense competing for light with the algae, the phytoplankton that's in the water. So you can effectively kind of reduce by a bottom-up effect, reduce phytoplankton simply because they're competing and they're able to shade the water column. Whether, you know, but they're also, depending on the type of lake you have, if you have natural vegetation that also competes for nutrients. So, you know, they may be competing with, you know, the nutrient side control. So it's hard to say, you know, it's very much a lake specific phenomenon if, you know, and what your goals are for the lake. So if they're beautiful and you like them, it really is up to individual lake associations to see if, you know, what are the benefits and costs for having lily pads.
And
Lauren, big things is that they you know, they're competing for nutrients as well as light with phytoplankton.
And Lauren, Beth has noted that for those who are interested in learning more about aquatic plants, such as lily pads, that PLEON is hosting a virtual workshop June 20th at 10 a.m. And you can go to the website for more registration details.
Thank you.